Hello, thanks so much for coming to our poster called Bridging Lab Science and Public Health with Wastewater-Based Epidemiology. I, as my co-authors here, are all part of the Molecular Monitoring Lab and in the Department of Cell and Molecular Biology at Grand Valley State University. One of the things that we're particularly proud of is the extent of our collaborations across Kent County. We work particularly closely with Kent County Health Department down there in the lower right corner, and also at the state level with Michigan Department of Health and Human Services. We are a part of a consortium of 20 odd labs across the state of Michigan that monitor wastewater for SARS-CoV-2. So we monitor from 36 sites across Kent County. The samples that we receive from these sites are processed at the Grand Valley State University campus, and then we analyze the extracts with digital droplet PCR housed at Kent County Health Department. So already you can see the collaboration between two different entities in Kent County. So since this project began in June 2021, we have analyzed more than 4,200 samples, and the analysis entails RNA extraction from wastewater and then reporting of basically three different things. One of them is the SARS-CoV-2 level, which has to be reported to the state level within 72 hours of receiving the samples. Another variable we have to report is the presence of variants. And the last one is the fecal content. Our workflow entails our partners grabbing samples. This can be from wastewater treatment plants, but we also contract with plumbers to grab samples from manholes. The wastewater sample is subject to an extraction protocol entailing precipitation and RNA extraction. And then once we get a very small volume of extract, we can quantify the amount of pathogen in there by taking it to our partners at Kent County Health Department and analyzing it with digital droplet PCR there. And then the results of that come back to our lab for analysis. And reporting. I'd like to highlight a couple of things of our project that make it especially powerful. In addition to the large number of sites that we have and the large county that we cover, we monitor fecal content using a fecal content indicator called peppered mild mottled virus. It is a ubiquitous RNA virus present in feces, and it is thus a very useful tool potentially for normalization of fecal content in our samples. You can imagine that the amount of fecal content could vary, and we do not want to erroneously ascribe an increase or a decrease in pathogen to what is actually an increase or decrease just in feces. So PMMOV is very useful for that. It also is a very useful tool for quality control, as you can see in the graph here, which shows a treatment plant result. You can see the PMMOV content as a function of time, and there's an outlier there right at the upper right, and we actually look at that and say that sample needs to be re-extracted. In addition, we do variant sequencing by a relatively novel method of amplicon sequencing, where we take samples with high COVID count, and we subject them to a nested PCR to amplify the receptor binding domain of the spike protein and send that off for sequencing. That domain of the spike protein contains a lot of diagnostic mutations for various variants. So those are two highlights of our program, that specific highlights that are sort of layered on top of the large scope of our project that involves a lot of community partners. And so it's one of the things couple of the things we're very proud of. So thank you very much for coming by and seeing my talk.